So now we're going to move on to talking more about human genetics. So one of the big things about human genetics is we can't do simple experiments like crossbreeding like we do in plants. Okay, so one of the things that can be used instead in humans is that you have these things called family pedigrees. And you can kind of look at your parents and your parents' parents to kind of look at how different traits are inherited and things like that. Now, I'm not going to go into much detail about those, but I wanted to give you an example of how you could use this in your everyday life. So I'm going to talk about my eye color specifically. Okay, and so I have kind of hazel-like eyes, but we're just going to consider them brown because eye color is kind of complicated, but in this situation, we're just going to call them brown. So, and this will work. Now, my mom has brown eyes. My dad has blue eyes. Okay? And in eye color, brown is dominant to blue. Okay, so this is true. And I won't go into too much detail right now about this, but brown is a dominant eye color over blue, which means that if we have, a heterozygote with we're going to use the letter B, so we'll say big B is brown, little b is blue. Okay, so big B would be brown. Big B, little b is going to be brown. And little b would be blue. Okay, so brown is dominant over that. So what we know is that for sure my dad has blue eyes. So he has to be this little b. Okay, and my mom, I don't actually know what her genotype is. I know that she has a big B because she has brown eyes, but I have no idea what the second letter is, okay? And another thing is my mom was adopted, so we don't really know what she's been able to find her genetic mother's eye color, but she doesn't know what her genetic father's eye color looks like. So we don't really have any idea what he actually had, okay? So we know that for me, so I have brown. Okay, so we're gonna say that I have B something. Now in this example, what I can use is my parents, what I know about them to decide what my genotype is. And I can say without any doubt that my genotype is actually gonna be big B, little b, okay? And the reason why I can know this for certain is because my dad, and we're assuming that he is really my dad. There's no drama in that case that we know of. My dad only has little b's that he could give to me. Okay, so there's no other option. So he had to have given me a little b, and so my eye color could be big B, little b. Okay, so this is how it's possible for two individuals that have brown eyes. Okay, so if we have, and this may have happened to you. Okay, you can have two individuals with brown eyes, but if they're both heterozygotes, okay, so this is their cross, so they both have brown, but they can end up having a blue-eyed baby, okay? Because what happens is, this is the different alleles and the gametes, so you can have big B, big B, big B, little b, big B, little b, and little b, little b. Okay, so neither of these individuals has blue eyes, but they're carriers of the blue eye allele, and therefore they can have a blue-eyed baby. So it is possible. So this is kind of a way that you can use human genetics and talking about them. Okay, so what I wanted to mention a little more about kind of human genetics is that a lot of times when we talk about traits as being dominant, I want to make sure that you understand that that doesn't mean that they're going to be dominant in the population, okay? So I'm going to write this out. So a trait that is dominant, okay, so for example, brown eyes over blue, does not imply that it is either normal or more common. Now, in the case of brown eyes are completely normal, and they are also more common, but there are other examples that are not, okay? So there are certain times where we have a dominant allele that is not common and is actually less common than a recessive. Okay, and so I'm going to give you an example in a second, but this basically, when we talk about dominance, this means dominance means that a heterozygous genotype is the dominant phenotype. So it's just basically saying that if we say it's dominant, that just means that if it's present, it will be showing up in the phenotype. Okay, so one really good example of a dominant phenotype that is not a well, I'm not gonna say it's not normal, but it's not common, is something called polydactyly. And what polydactyly is instead of having 5 fingers and 5 toes, you have an additional one. Okay, you can look this up. Apparently cats, it happens in cats quite a bit, but that would mean that if you have the trait or the allele for polydactyly, so for example, we'll just say that this one is, so capital D is polydactyly, and lowercase d is kind of your normal 5 fingers and toes. Most individuals in the population are going to have little d. So it's very rare for you to find someone that actually has an additional finger like that, other than, you know, the guy in Princess Bride. But most individuals have this, and so, but the thing is saying that we still consider polydactyly a dominant trait, because if it shows up, okay, so if you do have an individual that has the trait and they marry someone or just have a baby with someone, that individual could have offspring that will have kind of polydactyly. So it's definitely possible for this to be present, and it will be dominant if it is there, okay? But it's just a really rare allele. Well, the better term to say is, instead of saying like a dominant trait, what we will sometimes say is wild type. And what wild type means is that it's the one you see the most in nature. Okay, so those phenotypes most often in nature. So we would say normal 5 fingers and toes per hand and foot is the wild type for this particular trait. Okay, polydactyly is the dominant trait, but is not the wild type. Okay, so there are a couple human traits that are actually controlled by single genes, and we usually call these Mendelian traits, and we will get to look at these a lot in lab. Controlled by a single gene. Okay, and so there are a couple different traits. So for example, we'll talk about some that are determined by a single gene, but the majority of human traits are not easy and very simple, and they're much more complex. Okay, so I'm going to say that the majority of human characters have complex and poorly understood genetic basis. Okay, so lots of genetics we don't know much about. Okay, so for example, we really don't know a lot about eye color or hair color. Those are really complex. they're also not really that important in the grand scheme of things. It doesn't really matter that you have green eyes versus hazel eyes versus blue eyes versus brown eyes. People don't study that as much because there isn't really a therapeutic necessity. We also don't really need to know what color hair you have, but if there are traits that are very complex that are important to human health, then we do actually study them. But some things are very complicated, okay? And when they're very complex and poorly understood, those ones usually, they do not follow Mendel's laws that we saw. So law of independent assortment, law of segregation are not going to work. Okay, so don't follow Mendel's laws. However, there are a couple traits, and we'll talk about most of them in lab, that actually follow Mendel's laws, okay? And they can be applied to human traits that are determined by a single gene. Okay, so for example, a widow's peak. Okay, so kind of this is the hairline. So if you have a hairline in the widow's peak, that would be, this is determined by a single gene. Okay, and widow's peak is dominant over kind of the flat, straight hairline. Attached earlobes for your ear. So mind my drawing. So depending on how your ear attaches to your actual face, so sometimes you can have like these big, you know, kind of an earlobe that doesn't attach down here, and then other ones kind of attach directly. So unattached earlobes is a dominant trait, so you kind of have that earlobe part. Other individuals that have their earlobe that actually kind of connects directly to the side of their face, that is going to be the recessive trait. Okay, so those we'll talk about in lab quite a bit, and it's kind of fun to see which of the different traits that you have. Now, some of the things that are really studied well are going to be recessive disorders in humans. Okay, and so many, so most genetic mutations that cause medical disorders are recessive. Okay, and so what this means is that individuals that have the recessive allele but don't have the actual disease are going to be called carriers. Okay, so individuals who have the recessive, so basically the heterozygotes have the recessive allele but appear normal, are what we call carriers, okay, of the disorder. Okay, so for example, albinism, so being albino, is a recessive disorder, okay? If you have capital A, you have normal pigment. If you have capital A, lowercase a, this is the heterozygote. This is a normal pigmented, so I'm going to not rewrite that, but they're a carrier. And if you have little a, you are albino. Okay, so what can happen is you can have parents that are carriers. So if we have two individuals and they're not albino themselves, okay, they then have offspring, there is going to be a 25% chance that they will have an albino offspring. Okay, so you can do the actual cross. And this individual right here, so that would be a 25% chance of having an albino child. Okay, so it's 1/4. So there's 4 up here and one of them, so 1/4 is going to be 25% chance. Okay, this individual would have normal pigment and is not a carrier. 2 of these offspring could be carriers, and then the last one would be an albino. So recessive disorders are fairly common. However, there are dominant disorders. Okay, and so these ones are a little more scary because you only need one faulty allele to have the actual disease. Okay, so for example, there is a type of dwarfism. So it's called achondroplasia. Okay, so it causes dwarfism, so short stature. Okay, this is a dominant disorder, and so when you have the allele that carries the dwarfism, the dominant, so homozygous is dominant. What happens is this causes the death of embryo. So having 2 of the bad alleles is basically makes it so that a baby will not survive at all. A big A, little a will have dwarfism. Okay, and then little a would be then the kind of normal, quote unquote, stature. Okay, so this is what happens when you have a dominant disorder, because you just need one copy of the allele that's giving the dwarfism for it to show up. Now, what happens is that in this case, it's very high likelihood that you will pass on the dwarfism to, or any type of dominant disorder, to pass on the trait to your offspring. Okay, so if we have kind of a normal, let's just say a normal woman and a dwarf man, sorry, I'm switching letters on you. Let's say AA, and they have babies. So we have AA, little a. What happens is that 50% of their children are likely to have dwarfism. Okay, so 50-50 chance that they will have dwarfism when you have a dominant disorder. Okay, dominant disorders are less common, but more lethal. Okay, one really good example of a dominant disorder is called Huntington's disease. And what happens with Huntington's disease is that there is neurodegeneration, which is like the nervous system starts to degenerate. The problem with this Huntington's disease is that it doesn't start to show until after childbearing ages. So individuals that have this will have had children already and not know that they actually have the disease. And so when you have an individual with Huntington's disease, they have a 50-50 chance that they will pass it on to their offspring. So it's a very high probability, and this is a deadly disease. These individuals do not live for very long after they start to progress into the disease. The reason why it's scary is because, again, these individuals will seem like they have nothing wrong with them until after they've had babies. And so it's individuals that know that this runs in the family often will get genetic tested very early on, so they know whether or not they should have babies or if they need to kind of go about it at a different way, because you wouldn't want to give this to a child. Okay, and there's lots of different ways for genetic testing that we can do these days to kind of figure that out. Now, the last major things I want to talk to you guys about is kind of variation on Mendel's laws. Variation on Mendel's laws. Okay, and so these are going to be important because there are sometimes when we have these patterns of inheritance that do not coincide or kind of work well with Mendel's laws. Okay, and so the first one that we're going to talk about is called incomplete dominance. And so in incomplete dominance, what happens is that the F1 hybrids have an appearance, between the parental phenotypes. Okay, and so the best way to kind of represent this is just to show you what this would look like in flowers. So I think flower is a really good example of this. So what happens is if you do a cross between a pure breeding red flower and a pure breeding white flower, and that initial cross produces a bunch of pink flowers, you need to be triggered right away and think this is incomplete dominance. Incomplete dominance, the main word I want you to think about when you see this one is blending, okay? So think of it as red paint and white paint. If you put those two together, when they come together, they produce pink. So there is a blending of the two different alleles coming together. Okay, so it's incomplete because red is not overshadowing white. White is having an influence on the actual F1 generation. So there's a blending of this situation. Now, for a human example, there is this kind of disease that happens with cholesterol, okay? And so a normal individual will be homozygous dominant, and they are able to make what we call these LDL receptors. So LDL is one type of cholesterol, and a normal cell will have these receptors that are on the cell that are able to recognize how much LDL there is and basically be able to regulate how much LDL is in the body, okay? If you're a heterozygote, what happens is that there's a mild disease and you do not have enough normal LDL receptors, okay? And so this is going to be kind of the intermediate. When you have the severe disease, you have absolutely no receptors. And so when we have kind of going between a normal amount of receptors and a no amount of receptors, and in between is a blending, so it's kind of halfway in between, we then know this is going to be incomplete dominance, okay? If this was a, if this normal was a kind of a dominance trait, then we would think that the heterozygote would also have the dominance trait, which would be normal LDL receptors, but they don't, because they're kind of halfway in between what's normal for the homozygous dominant and the homozygous recessive in this case. Okay, so that is incomplete dominance. So incomplete dominance, blending. The next type is called codominance, and this one is not blending. So no blending on this one. Okay, so in this case, the F1 hybrids have both phenotypes present. Okay, neither, oops, that didn't spell that. Masks the other. Okay, so no blending. So they're both present. Okay, the best example of this is blood. So blood types. Okay, and I'm going to change my pen. Let's go with red. Okay, so for blood types, there is codominance. Okay, so we have what we call an ABO blood groups. Okay, and what it turns out is there are 3 alleles instead of the normal 2 for a single gene. Okay, and these 3 alleles will come together and produce 4 phenotypes. Okay, so you can have A blood, you can have B blood, you can have AB blood, or you can have O. Okay, and what these A and Bs are actually referring to are carbohydrates that are on the actual surface of a red blood cell, okay? So the letters refer to two carbohydrates designated A and B. Okay, and these are found on surface of red blood cells. Okay, so this is not at all what they actually look like, but I think it helps illustrate what's going on here. So here are 4 red blood cells. And so for A blood, what you would have is these little A carbohydrates on the outside of the cell. Okay, for B blood, you'd have B carbohydrates. For AB, you would have A and B. Okay, and so because this is the codominant one, so instead of having a blending like a U letter, we have both of them present and unaltered. They're just both going to be there. Okay, and our last group, O has no carbohydrates on the outside. So it doesn't have A's or B's. So blood groups are a little bit more complicated. And so I'm going to kind of show you what the genotypes actually look like for this, because it will come up in lab. And so when you have the 3 alleles, you have 6 possible genotypes. And first of all, this is going to be a little different, but we're going to use the letter I's kind of as part of this, okay? And we're also going to use the letters A and B. So basically, I'm going to start writing these. Okay, so if you're an A-type blood, you can either have big A on these I's, or you can be a big A, little I, which means that you're this is the heterozygote, okay? For B, you can have 2 capital I's with the B's on there, or you can be like this, okay? For AB, there's only one possible combination that can make an AB, like that. And then if you have O blood type, this is what we would call your genotype, okay? And so these two both would have A blood type. These both have B blood type. This one is AB blood type, and this is O, okay? And so this is all very complicated, but the main thing that we're codominant is this one right here. So we have codominant situation when you have kind of the A and the B. Okay, so A and B are codominant. A and B codominant. And little I is going to represent the recessive for everything. Okay, so if you have a little I, it does not affect the phenotype. Okay, people that have AB will have both A carbohydrate and B carbohydrate. Now, the reason why it's really important to understand this and to know what your blood type is that if you are an individual and you have A blood type and they give you a donation of blood from someone that has B or AB, it will cause an allergic reaction and will actually potentially kill the individual. And this has happened before they kind of understood how this all worked. O blood type is a universal donor. It has 0 carbohydrates on the outside of its red blood cell. And so basically the immune system of the recipient doesn't recognize it as being foreign. But if you give a person who has A blood type, B blood type, it will recognize those B's as being foreign and it will attack them. Okay, and so this is why it's very important to have the right kind of blood given to you. All right. homologous chromosomes, you can sometimes have something that's called a linked gene. Okay, and so what this is that you have genes that are located near each other on the same chromosome. Okay, and what happens is if they're close enough, they will travel together. So tend to travel together during meiosis and fertilization. Okay? And so what happens is we previously were talking about something called independent assortment, where two traits didn't affect each other. Well, this is what's going to happen when traits do affect each other. So linked genes are often inherited as a set, and therefore do not follow Mendel's law of independent assortment. Okay, so just to show you kind of what this looks like, if we have kind of mom's DNA, dad's DNA, and then I'm going to draw on here, we have this blue gene, blue gene, and orange gene, and they are very, very closely located on the same chromosome, okay? What's going to happen is that during crossing over, it's unlikely that they're going to get split apart from each other. So they are going to actually stay linked together on their, kind of in the way that they are. Okay, so unlikely crossing over. Okay, remember crossing over is when we actually exchange big chunks of the actual DNA on homologous chromosomes. Crossing over would split these apart. Okay, so these would not behave in the same way that other traits like Mendel's laws would, because they are actually linked together. All right, and last but not least, we're just going to mention this. Make sure. I want to talk a little bit about sex-linked genes. So if you remember, we have 23 chromosomes, and 22 of them are what we call autosomes, which means that they're basically the same chromosome. But we have one set of chromosomes called sex chromosomes. Okay, so that all adds up to 23. So we can have XX and XY. And there are certain genes that are located usually on the X chromosome that are linked to different diseases, okay? So a sex-linked gene is a gene located on a sex chromosome, okay? And examples of this are color blindness and hemophilia. Basically, what happens is that you have kind of this unusual pattern, because what's going to happen is you have individuals that if the trait is on the X and you have a male, they don't have they're not the same chromosome or genes on the Y chromosome, okay? For autosomes, all the same genes are on each chromosome. For the X and the Y, these have different chromosome or different genes because they are not the same chromosome. Okay, and so in women, you often will have XX, where if you have a faulty gene on one of your X's, you have another backup gene. But for men, they don't have a backup gene because the Y chromosome does not carry the same genes as that's on the X chromosome. Okay, and so for example, one disease, color blindness, is a sex-linked trait, okay? And it affects men much more than it does women. So what I'm gonna do right here is I'm gonna say that N equals normal color vision and little n equals colorblind. Okay, and because we're talking about sex-linked traits, we have to have the X's and the XY, okay? So if we have a male and a woman, male and female, and we're gonna say that the woman is a carrier for color blindness, so she has a capital N and a lowercase n, and we're gonna say that the man has color blindness, okay? So we're gonna say X, his N for his color blindness is on his X, his Y does not carry the colorblind gene because it doesn't carry any of that same stuff. And so what you would do is you could do a cross between this, and it's very similar, just kind of looks different because you're using different letters. But if we put the man right here, and we have the woman right here, we can have, in this case, let's see, X big N, Y, X little n, Y. Okay, so the man is going to have a little lowercase n, so it has color blindness, and this woman is a carrier, and so she can end up creating, because both of them they can have a daughter or a son that are actually going to be colorblind. Okay, and so the way that these actually come out is a little bit different and do not follow Mendel's traits. Okay. We will talk more about this when we get to lab, and I will kind of help you more with these crosses. Okay, and if you have any questions, please email me, let me know, and I will try to help you as best I can. Thank you.